This is part 4 of the Chapter 6 lecture notes on metabolism, and we were finishing up on photosynthesis, the first part called the light reactions. This, again, is a picture of a single thylakoid. You can see that the membrane of the thylakoid has a double layer of phospholipids, just like the plasma membrane. And we talked about the steps of the light reactions. Begin with photosystem 2, which is right here. An electron is excited by sunlight. It bounces around the various pigment molecules in the antenna assembly until it arrives at 2 chlorophyll A molecules called the reaction center, and then it is transported to the electron acceptor. And from the electron acceptor, it is transported to the electron transport chain. And the electron transport chain uses chemiosmosis to create ATP. That is by generating a hydrogen ion concentration inside of the thylakoid membrane. And as the hydrogen ion diffuses back across the membrane from its high to low concentration, ATP synthase uses that flow of hydrogen ions, to add a phosphate to ADP to create ATP. So photosystem 2 and the electron transport chain create ATP, and then that same electron moves on to photosystem 1 and undergoes the same things. Sunlight is absorbed, the electron arrives at the reaction center A, chlorophyll A molecules, and then the electron acceptor molecule, and then it is passed along these electron carriers, and that energy is used to create NADPH. So the products of the light reactions are ATP and NADPH, and they are going to go on and be the power for the Calvin cycle, or the light independent reaction. So the ATP and the NADPH that are accumulated during the light dependent reactions are used as fuel by the Calvin cycle reactions. And the Calvin cycle reactions occur in the stroma, whereas the light dependent reactions occurred on the thylakoid membrane. So the Calvin cycle reactions are shown here. Basically, carbon dioxide is the carbon source, and that carbon dioxide is combined with RuBP, and RuBP contains 6 carbons. It is combined with RuBP, and then that's the 6 carbon, I'm sorry, I said that wrong. I'm so sorry. I'm not going to re-record this, but RuBP has 5 carbons. Then, when you add the carbon dioxide, the molecule that's formed has 6 carbons, but it immediately splits into 2 molecules of 3-PGA. And each of those have 3 carbons each. And ATP is used, and the NADPH is used during this next step, which is the generation of G3P. And we call this step reduction. The first step is called carbon fixation. The second step is reduction, and you can see why, because what is contained in that hydrogen atom is a hydrogen ion and an electron, and it gives that electron to the G3P to form the G3P, gives it to 3-PGA to form G3P. The G3P is the molecule that's used for food by the plant, but some G3Ps are recycled, and ATP is used to recycle them back into RuBP. This shows you the stages a little bit more clearly. The first stage is carbon fixation, and that occurs when RuBP, which has 5 carbons, you can see them right here, 1, 2, 3, 4, 5. The 5 carbon RuBP combines with 1 carbon from carbon dioxide and produces a 6-carbon molecule, which is split into 2 3-carbon molecules. This is called carbon fixation, uses the enzyme ribisco, and it has been discovered that this enzyme is the enzyme in the greatest concentration of all enzymes in the entire world, which lets you know how important photosynthesis is to living things. The enzyme ribisco is the one we have the most of. If you looked at all the enzymes in all organisms on the earth, ribisco would have the highest number, the highest amount. The reduction stage is when ATP and NADPH are used to reduce 3-PGA into G3P or GA3. GA3P and G3P are the same thing. You go back to this page, it was written as G3P. Same thing, GA3P is glyceraldehyde 3-phosphate. But that one turn of the Calvin cycle actually uses 3 molecules of carbon dioxide. And so instead of producing 2 3-PGAs, it produces 6 molecules of 3-PGA. Instead of producing 2, it produces 6 because 3 times 2 is 6. Those 6 molecules are used to produce 6 molecules of GA3P, but only one of those molecules of GA3P can be used to produce food. 5 of them must be recycled into stage 3, which is the regeneration of RUBP, which is a 5-carbon molecule. All right, now we're moving on to cellular respiration, and there are 4 stages in cellular respiration. Glycolysis is the first. Cellular respiration occurs in the mitochondria, whereas photosynthesis occurred in the chloroplast of plants. Now, all cells, including plant cells, contain mitochondria, and all cells undergo cellular respiration, except for some unicellular organisms that are anaerobic, and they don't undergo cellular respiration. But almost all cells, including plant cells, animal cells, and fungi, undergo cellular respiration, and the first stage is glycolysis. The second stage is I just call it the PrEP reaction. The third stage is the citric acid cycle, and the fourth stage is the electron transport chain. So glycolysis is the first pathway used, and it's the breakdown of glucose. Glyco means glucose, lysis means breakdown. So what we're going to focus on really is what is produced. So in the end, at the end of glycolysis, you have 2 ATP molecules. So a net gain of 2 ATP molecules, and then a net gain of 2 NADH molecules, and then 2 pyruvate molecules. And you can see that here. Glucose is a 6-carbon molecule. During glycolysis, it is broken into 2 3-carbon molecules called pyruvate or pyruvic acid. And as a result of this reaction, 2 ATP molecules are produced and 2 NADH molecules are produced. So glycolysis produces 2 ATPs of the 36 or 38 that are going to be produced by this glucose molecule. Glycolysis occurs in the cytoplasm of the cell. It does not occur in the mitochondria. And you don't have to get all caught up in the individual steps of glycolysis. But the first half of glycolysis uses 2 ATP molecules, and the second half forms 4 ATP molecules. And that way, 4 take away 2 is 2 overall ATP molecules that are produced by glycolysis. So, and this is showing you all the stages of glycolysis, and that the end product, the reactant is glucose, which contains 6 carbons, and the end product is pyruvate, which contains 3 carbons. It's 2 molecules of pyruvate, which contain 3 carbons. And as well as that, 2 ATPs are produced and 2 NADHs are produced. So these are the overall products of glycolysis: 2 ATPs, 2 NADHs, and 2 pyruvates. Then the second step is called the oxidation of pyruvate, or pyruvate oxidation. And this removes a carbon from the pyruvate. And since it had 3 carbons, it's going to form into a 2-carbon molecule. And that 2-carbon molecule is the acetyl group, which attaches itself to coenzyme A. So we call it acetyl-CoA. So the pyruvate, which has 3 carbons, is converted into a 2-carbon acetyl group that's attached to coenzyme A. Okay, in this process, carbon dioxide is given off, because that's where the carbon dioxide has 1 carbon, and that's how the pyruvate loses its carbon, is as carbon dioxide. And 2 more NADHs are produced, but no ATPs are produced. So we end up, after the pyruvate oxidation, which is shown right here, the pyruvate is converted into acetyl-CoA, which is a 2-carbon molecule. And it would end up being converted into 2 of these because there were 2 pyruvates in the beginning, produced by the glycolysis step. And the acetyl-CoA, 2 of those are produced, 2 NADHs are produced, and 2 carbon dioxides are produced during the oxidation of pyruvate. And this step does occur in the mitochondria, not the cytoplasm. All right. Then the next step in cellular respiration is the citric acid cycle, which is sometimes called the Krebs cycle, for the scientists who discovered it. The citric acid cycle begins when the 2 acetyl coenzyme A molecules enter the citric acid cycle. And it's a series of reactions that first builds back up the number of carbons in the molecule and then breaks them down 1 carbon at a time. The purpose is because these reactions produce a large amount of NADH and FADH2. Now, these are carrier molecules. They contain hydrogen on the end, and these hydrogens are carriers of hydrogen ions and electrons. And we've already talked about how hydrogen ions and electrons are used during chemiosmosis to produce ATP. Now, the citric acid cycle will produce either 1 molecule of GTP or ATP. They're both used for energy by cells per turn. But there are 2 turns of the citric acid cycle because there were 2 acetyl coenzyme A's that were produced in the last step, the oxidation of pyruvate. So the 2 acetyl-CoAs means there are going to be 2 turns of the citric acid cycle. And the 2 turns of the citric acid cycle result in, and we can count them from here, okay? The first step is that the 2-carbon acetyl-CoA combines with the 4-carbon oxaloacetate, producing a 6-carbon citric acid or citrate. And as that citrate is broken down, it is used to produce NADH. I see 3 of those. And FADH, 2. I see 1 of those. And it is used to produce 1 GTP or ATP directly. So if we want to write down the overall products of the citric acid cycle, the overall products of the citric acid cycle are total, since we have 1, 2, 3 NADHs and there are 2 turns, we get 6 NADH molecules. And we get 2 total FADH2 molecules. And another thing that's given off in the citric acid cycle is carbon dioxide. And you see that here and here.